report that the Agriculture Innovation Board put out. And today we're going to just hear about that. We're going to hear about pollinators. We're going to hear about bees and maybe a little bit more from an expert. We're not experts, so we'll know a little bit more after we hear the testimony this morning. So good morning again. If you want to, let's introduce ourselves first. So I'm David Durfee. I chair the committee. I represent the towns of Shaftesbury and Sunderland and Glastonbury in Bennington County. And why don't we just go around?
Roger, I'm a representative Orange 3, which is Williamstown and Chelsea.
I'm Representative Jed Lipsky. I represent Lamoille 1 District, Stowe.
Representative Charles Wilson of Caledonia 3, which is Lindenville, Sheffield, Wheelock, Newark, and Sutton.
David Templeman. I'm Representative for Orleans 3, which includes Barton, Brownington, and Westmore.
And I'm Esme Cole. I represent Hartford, which is 3 through 6.
Heather Supernut, Vice Chair, Windsor 4, Barnard, Pomfret, and Bridgewater.
And I'm Josie Levitt on Zoom. I represent Grand Isle, all of Grand Isle County, and a sliver of West Milton.
And we have, I think, joining us when they have the chance, Representative Pearl and Representative O'Brien. Representative Rice is here today, and he'll be back in a few minutes, I think. So let's turn it over to you, Dr. Elder, please. If you wouldn't mind introducing yourself for the record, and then we'd be delighted to hear whatever you have to share with us.
My name is Samantha Alter from the University of Vermont. So I've been asked to present a little bit of education around pollinator health in Vermont and some of the pesticide monitoring work that I've been doing in my lab. I've been asked to say the views expressed are my own and do not imply endorsement by the university.
Okay.
So introduction about myself. Many of you I haven't met before. I got my PhD at the University of Vermont, where I studied how diseases are spilling over, particularly viruses are spilling over from managed honeybees into wild bee populations. And then I stayed on, and now I'm a research assistant professor at UVM in the Plant and Soil Science Department, and I run the Vermont Lab, which is housed in Jeffords. We got a great team that helps us out with our field work and our lab work. We run education programs where we teach beekeeping classes to the general public and students at UVM at the horticultural farm. We have a diagnostic lab where beekeepers can send us samples of their bees, and we give them reports for free doing pest and pathogen diagnostics to help with their management decisions. I also, since 2015, I've managed Vermont's involvement with the USDA APHIS National Honeybee Survey, which is a nationwide effort to gather baseline data on bee disease and pathogens. And then lastly, we just have a number of different research projects that we are pursuing in our lab, and one of which I'll be talking to you a little bit about today is some of the Vermont pesticide monitoring that we started a few years ago. So the outline of today's talk, I'm going to be talking about the pollinator trends, what we're seeing for honeybees, which are managed bees, and wild bees, how their populations look here in Vermont. Talking about introducing you to threats to bee populations. Neonicotinoids is a class of pesticides that have been in the limelight as of recently, and so we're going to talk a little bit about what neonicotinoids are. I'm going to share with you some of the pesticide data that we've been collecting, talk about how neonic pollinators are exposed to neonics, and then a little bit more about some of the data that we've been seeing about how neonicotinoids as a tool may or may not be impacting crop yields and how farmers can be supported. We've been doing a lot of research on that as of lately. So through their pollination services, bees provide somewhere in the realm of $235 to $577 to the agricultural economy. So this is like the why do we care about pollinators?
And that's nationally?
Yes. They are also critical for the reproduction of our wild plants. And I think oftentimes when we've all sort of heard save the bees, or bees, the bees are in trouble. Oftentimes what we're thinking about in our mind is this iconic species, which is the European honeybee, Apis mellifera. And this is one single species of bee. And it's actually non-native to the US. It was brought here by colonists for honey production. And they're managed as agricultural livestock animals. That's what they're considered here in the US, as a non-native introduced species of bee. They're incredible. They're, we can farm them. Like, this is an insect that we farm, which is really, really cool. And they do really well in these little boxes that we can ship all across the United States to pollinate our food crops that we grow in big monocultures, right? Where we need a lot of pollination in one place at a short period of time. So we can bring these bees and drop them in, and they provide those pollination services for food crops. So looking a little bit at Vermont's honeybee industry, according to the Agency of Ag, just in 2023, there was about 17,000 registered hives in Vermont, half of which are migratory. And those are the ones that I mentioned earlier, that they're being trucked around and being mostly propagated in the south. And then they get trucked in for the summer for a short period of time. And that temporarily doubles the number of hives, more or less, that are registered in the summer. And then they leave again and go back down south where they get propagated. Beekeepers as a whole across the nation, this isn't a secret, they're experiencing very high colony losses. And beekeepers fortunately are really good at making more bees. And this is why over time, we haven't seen the number of colonies, of honeybee colonies, decrease over time, because beekeepers can make splits. If they have a really healthy hive, they can make that hive into 2 or even maybe 3. But making these sort of splits to make up for their losses cuts into their profits and their ability to make honey, and so on and so forth.
Question, and is it okay if we jump in with a
Please.
question?
Yeah.
Is the European honeybee a single species that is the one critter that's used for all of our pollination management?
There are a couple other managed pollinators that are like managed bumblebees and alfalfa leafcutter bees, mason bees, but the European honeybee is the most important managed crop pollinator. So to get a better understanding of honeybee health, the best metric for that is colony loss. So this means the percentage or the number of colonies that beekeepers might lose in a given season or during the winter. And always, beekeepers have always, as farmers, beekeepers always expect to lose a certain proportion of their hives every year. But this has increased over time to a point that beekeepers feel, and surveys have shown that they feel that these numbers are not sustainable. So the recent report from the Agency of Ag reported winter colony loss. So this is the percentage of colonies that beekeepers lose during the winter months, not the whole year. And beekeepers have always lost bees during the winter months, but now we're seeing beekeepers losing colonies throughout the entire season. And so 28% is what the agency is reporting for winter losses for Vermont beekeepers. And if we look to other estimates of colony loss, the most reputable source of this is the Bee Informed Partnership surveys that are a nationwide colony loss survey that gets put out. They calculate annual loss in Vermont for the same year to be 71%. So we can see there's a range here that's being reported. But if we look to some of the historical, not just 1 year, because it's important to look at a wide variety of range of years to see how bees are faring or beekeepers are faring over the course of the season, we can see that since 2010, Vermont lost a range of either somewhere between 15 to 85% of their colonies each year. And I've got these QR codes throughout, so that way you can, if you feel comfortable scanning it, you can go and see the data yourself and explore. They have a really great user interface where you can click on the state and see how it compares to other states and understand how these calculations were made. So that's a little bit about the managed honeybees and the colony losses that beekeepers, these farmers of bees, that's what they're experiencing. But as I mentioned before, they're not the only bee species that we're concerned about. There's over 350 native bee species here in Vermont. And so to understand a little bit about how those populations are doing, in 2018, Leif Richardson and other colleagues looked at just bumblebees in particular, because we have really good estimates of historic collections of bumblebees, because naturalists were really interested in this fuzzy charismatic species. So we can compare historic and modern day collections, and found that there's been a loss of 28% of historical diversity just within the bumblebees. But But very recently, the Vermont Center for Eco Studies put together this really awesome report, The State of Vermont's Wild Bees. And I'd argue that because of this work, Vermont has a better understanding of our native bee populations than most other states. And what they found is that over 30% of Vermont's native bee species are ranked as critically imperiled or imperiled, and 55 of Vermont's 350 species are in need of conservation action. And so these are the species that we're not farming. These are the species that you can't make more of, whereas honeybees are an agricultural livestock animal following very closely. And I think folks feel that the honeybee is sort of this canary in the coal mine. If things are happening to the honeybee, then it's an indicator of broader implications for the ecosystem. And thanks to VCE, we are seeing that those trends in pollinator losses are playing out for many species here in the state and elsewhere. Hi, morning. So what's causing the issues with bees? Scientists are in agreement that it's not just one thing, it's an interaction of multitude of stressors. And I'm going to briefly discuss these before I then jump into some of the specifics of pesticides and neonics. We know that climate change, of course, is an issue. The introduction of pests and pathogens, the loss of clean forage and habitat, and then pesticide exposure. So briefly on some of these other threats. So climate change, of course, droughts, when it's a drought year, the flowers don't produce the nectar that bees need to eat. That's much of their food source. 75% of wild bees actually nest underground, and which I think is surprising for most people. And so you can imagine how floods might impact ground nesting bees. And it's going to be really interesting to see how these populations are going to be impacted after this past year of flooding. And there's some work being done at UVM on this topic. As temperatures have risen, we've seen a reduction in habitat for some species. Bumblebees like cooler climates, higher latitudes, and so bumblebees have lost a lot of habitat over the past years.
Representative Lipsky has a question.
I apologize for going back, but
That's okay.
you showed a chart and had one of those symbols in the corner
That's
that
right.
showed New Hampshire as the highest rate of loss of bee population. That one.
Yeah.
And right to the west of New Hampshire, it showed Vermont, which was similar to Iowa and Indiana, of quite severe colony loss. Have you analyzed why our region, were there any environmental or climate issues going on in northern New England that made us stand out in not a good way?
It's a great question. I would say that if you go and explore the data, you'll find that this isn't the same year after year. And so you'll see other states popping up as having high colony losses in any given year. So I wouldn't say the trend of Vermont or New Hampshire being the highest in colony losses is something that's consistent across years. But this particular year, this 21-22, we did see a high colony loss. I don't have an answer as to why this particular year. No, that's all right. No, happy to take questions. I adjusted my talk to accept questions, so I'm not going to go over, I don't think. So pests and pathogens are a huge issue for managed and wild bees. I spent a lot of time during my PhD studying this topic. I really have to mention that the Varroa mite, which is pictured here, is considered to be the #1 pest that beekeepers deal with. Now, the Varroa mite or Varroa destructor wasn't always here. It was brought here from Asia in the late 80s and changed how beekeepers keep bees. Beekeepers are spending most of their time trying to control this pest. The pest is, it's an ectoparasite, meaning it lives on the outside of the bee's body. It eats the hemolymph or the blood of the bee and the fat bodies of the bee, much like a tick on a mammal. And it also transmits viruses to the bee, kind of like how ticks might transmit Lyme disease. Similar to that.
Can you say the name of the
Yep, Varroa.
Varroa?
Yep, Varroa. Yep, and you'll hear a lot about Varroa if you're hearing from people about pollinators. Yes. Yeah. So beekeepers spend a lot of time trying to control this Varroa mite using different kinds of management practices, actually different kinds of pesticides inside their hives to try to kill a bug on a bug, which has been extremely challenging for the beekeeping industry. And we do know that other factors like, for example, reduction in nutrition problems or pesticide exposure can exacerbate the impacts that you see of this parasite. So we see an interaction effect here. So the habitat availability of clean forage, this is forage that has not been sprayed or provides routes of exposure for pollinators to come in contact with neonics, is also a big issue. With agricultural intensification, research has found that it diminishes the pollination that native bees can provide by 3 to 6-fold. And we know that native bees can provide really great pollination. We've seen this work from folks from out of UVM looking at how native bees are great pollinators of blueberry crops. And this sort of reduction in native bee population then puts reliance on this agricultural livestock animal, the honeybee, to provide that pollination service for us. If that makes sense. So as we sort of create areas that are, looks a little bit more like this than with hedgerows that provides habitat for native bees, we become more reliant on this agricultural livestock animal to be shipped in to provide those pollination services for the monocrops. And this puts pressure on the beekeeping industry and so on and so forth.
So go ahead, Representative Templeman.
A question about habitat, for the 300 plus species that are native to Vermont. What is the ideal habitat for them? I mean, I'm thinking like fields of wildflowers,
Sure.
but we are a heavily forested state. And I can imagine the dark recesses of the forest don't allow wildflowers to propagate. So what is that ideal non-human environment look like?
Yep. That's a great point. So there are many specialist bees that have very specific needs, and that report from the Vermont Center for Eco Studies actually talks about all those kinds of different habitat types that are really important for some species. But I would say, if you're going to say it more generally, these kind of open habitats like fields, hay fields with flowering plants, where it lets the sunlight in that allows flowering plants to grow. But trees are also great resources of nectar and pollen for pollinators as well. Yeah.
So just going back to the point you were making here, absent managed hive, if I have an apple orchard, for example, there would still be some natural pollination taking place from the wild honeybee population, or the wild bee population,
Yep.
the wild pollinator population.
Yeah, great. Yes, as long as you've provided habitat for those wild bees,
Okay.
so that they have a place to live and that they're close by to your orchard.
Yeah.
Great questions. Okay, so talking a little bit about pesticides and how pollinators or bees or butterflies might be exposed to these all different kinds of pesticides. I'll start with, when I say pesticides, we're talking about herbicides, which kill plants, fungicides, which kill fungus, insecticides, which kill insects. But then also miticides are also a pesticide that kills the mites that beekeepers actually put inside their hive to try to combat that varroa mite parasite. But if we're talking about, insecticides and herbicides, or, those kinds of pesticides, these are the various ways in which bees might come in contact with them, either through direct exposure, which is probably the worst, they're just directly sprayed, residue contact where flower or resource might have been sprayed previously, and then a bee will visit that flower or resource after. A lot of solitary nesting bees or cavity nesting bees will use leaf tissue and soil to create nesting areas for themselves. And they'll take them back to their hive, their nest sites to raise their babies. And so if their nesting materials are contaminated, of course, that's a route of exposure. And then, as we mentioned before, 3/4 of native bees are nesting underground. And so in pesticides that are harmful to pollinators, persistent in the soil, that could be another way in which they can become exposed to those. So I borrowed this slide from Xerces, talking about not all pesticides are problematic for pollinators, but there are sort of things that you look out for that are considered to be red flags. And that's the how toxic, of course, how toxic they are to the pollinators, whether they're used a lot and if they move off site easily, how long they stick around, so the persistence. If they stick around for a long time, you can imagine that that results in more opportunities for exposure. And then the kinds of ways in which pollinators are exposed. And so with neonicotinoids, they sort of check all these boxes. And that's why, one of the reasons why they've been brought up a lot lately. And we've seen that New York has recognized some of these impacts of neonicotinoids with recently passing the Birds and Bees Protection Bill, where they phased out neonicotinoid pesticides. And I think this is sort of like led to, neonics have been discussed a lot in the pollinator community, but now this has sort of put them directly in everyone's view, especially up here in the Northeast. So neonicotinoid insecticides, a little bit of introduction about them. They are a class of systemic pesticide. The majority of their use is actually as a seed coating on the majority of row crops. So the state put out a report and found that the majority of over 99% of corn in the state is treated with, or is grown with neonic treated seeds. I think 35% of soy, and this is where the majority of neonics are being used. Other uses include foliar sprays, soil drenches, trunk injections, and then of course lots of fruit and veggie crops, turf grass, and ornamental plants. So the properties of neonics are they are considered by EPA to be highly toxic to many classes of insects, including pollinators. They're systemic, meaning that when the plant grows, it takes up these insecticides into the plant tissue, where it then can provide the plant with some benefit if a pest were to try to chomp on a leaf of that plant, right? But as being systemic, they have these really, really small molecules that make it highly water soluble and can move out throughout the environment if it doesn't end up in the target plant. And so a little bit about treated seeds and how these things work. So if you have a neonicotinoid treated seed, they look like this. They're colored to indicate to the farmer, the practitioner, that these are in fact coated or with insecticides. They're planted into the ground, and only about 2 to 3% of the insecticide is taken up into that target plant. And the rest of it, research has found over 90% actually don't end up in that target plant, but leaches out into the soil and waterways. And we've seen through research that they're persistent in the environment for years.
So this sounds like it might be significant that only a small number, 2-3% is taken up by the plant. Is that though, and would that be enough to
provide protection to them?
To accomplish the point, the purpose of having the seed treated? So
as I'll talk about later, the research is showing that these neonicotinoids provide little to no benefit to crop yields. And there's been trials done here in Vermont that have found it as well. And I don't know whether or not it's because of the small amount that's taken up into the plant, or if there's just not the pest pressure that would result in a problem. Yep.
Okay, thank you.
Sure. So the one thing that I want to mention about the systemic properties of neonics is that as they're taken up into the plant tissue, they're also expressed they would be expressed in the nectar and pollen of a plant as they're growing. So you can imagine this could be one way in which a pollinator could come in contact with that plant and then be exposed to these neonics. So the impacts of neonics are that they're impacts to pollinators with very, very small concentrations. They're highly toxic in tiny quantities. And there's these sublethal, so there's lethal exposures and there's sublethal exposures. And sublethal means that there are things that make the bees less able to forage or larvae to fight off disease, which then results in a dwindling of the bee population. They're sicker, and then that could eventually lead to death. But we've also seen a number of field realistic studies that have found neonics implicated in the reduction of wild bee density, solitary bee nesting, bumblebee growth and reproduction, the reduction of overwintering success, colony reproduction for both bees and wild bees, decreased survival, and immune response of honeybees. So at some point, if you would like a bibliography of all of the impacts of these neonics to pollinators, I'd be happy to provide that to you. So, I've been asked to testify for a number of bills in the past related to neonics. And at those meetings, their question kept coming up about, well, are the bees here in Vermont exposed to these insecticides? And at the time, this is, back when I was a grad student even, we didn't have much data on what bees were bringing back to the hives, what kinds of insecticides or pesticides they were exposed to. So then a few years back, we said, well, let's just start collecting the data. And so some of the data I'm sharing with you today, I actually just got back a couple weeks ago and just started analyzing, and this is the first time I'll be sharing it with anybody. So we started this pesticide monitoring program, and to understand how it works, you got to understand a little bit about bee biology, but they've got these concave little pockets on their legs, and as they're out in the world collecting, visiting flowers, collecting pollen from flowers, they take these pollen granules and stick them to their legs. And so as beekeepers or scientists, we can take advantage of the fact that these little bees are traveling throughout miles from their hive, sometimes 2, 3, 5 miles from their hive, and coming in contact with lots of different things out in the landscape. If we're able to sort of, as they come back to the hive, pull the pollen off their legs to get a snapshot of what that bee came in contact with as it flew out across the landscape. So they're really cool and being like bioindicators of what's out there. And a huge workforce, tens of thousands of little bodies that are going out doing sampling for us. And we're also able to understand not just what's out there in general, but like what the bees are bringing back to their hives that they're then going to feed to their larvae or their babies. And so we have these things called pollen traps, which are really cool. They're a series of either these little holes or a series of screens that as the bee flies back to their hive, you fit your hive with these pollen traps. And as the bee flies into the entrance and kind of navigate through these screens or little holes, the pollen gets scraped from their legs and lands into these little troughs. And so each little pollen load that you see here represents one of these little things from their leg. And they're all different colors because they're visiting a diversity of different flowers. So we've done that since 2021 and worked with a number of beekeepers out in the community, setting up pollen traps on their hives to see what their little bees are bringing back to their hives. And then we collect all these pollen samples and we send them out to Cornell, the Cornell Chemical Ecology Core Facility. I'm just going to mention broadly, it's $90 a sample, which is an incredible deal for doing this kind of work. The service is available to anybody, beekeepers, farmers, researchers. They do a 93 pesticide multi-residue screen. I've heard that they're going to be doing other things in the future. And there's equipment is very, very sensitive. And this is a really important thing to know because as I mentioned earlier, these pesticides are impactful in such tiny quantities. You need a really, really sensitive machine to find that small amount. And so the limits of detection in this case are less than 1 part per billion for most of the pesticides. So you can imagine if you're using equipment that tests at 10 part per billion, you're going to get false negatives for anything that's found at those lower levels, which are significant for pollinator health. So we're really lucky to have this right near us and that we have this resource for us. So in 2021, this is our first year of sampling, just more broadly, talking about all pesticides here, not just neonics, pesticides being, herbicides, fungicides, insecticides, right? Miticides as well. We started small, it's kind of expensive in some ways, $90 a sample. We looked at 16 samples and we were surprised to have 81 detections of 20 different kinds of pesticides, just in that small sample. Wow, these bees are coming in contact with the different things that could potentially all have synergistic impacts on their ability, on their health. So we continued that work in 2022. We had 18 bee collected pollen samples, found 89 detections, again, of 23 different pesticides, a similar amount. And for those of you who are curious about, did you find any of those neonics? About 21% of the pollen samples from those years, we found at least one of those highly toxic neonicotinoids, which you'll hear these names over and over again if you talk about neonics. Clothianidin, imidacloprid, and dimethoxam are those 3 big neonics that you'll hear about a lot, and the ones that are considered highly toxic. And so if we looked at the 21 data, this is old data, I'll get to the new stuff in a little bit. But if we looked at that 2021 data and classified the hits that we were getting for different pesticides based on their category, whether they were fungicides, herbicides, insecticides, neonic insecticides, miticides, or synergists across the season here. So these are the months May, June, July, August, September, and these are the percentage of detections for each of those categories. And what we found, we were interested in seeing that these neonics are appearing in May, a little bit in June, and then they're popping up again in August. So we're wondering, so what are the exposure routes that are leading to these, higher points when we're seeing these neonics? And I am not the first person to look into this. There's a lot of this out in the literature, but it was interesting to do this right in Vermont here. So talking a little bit about exposure routes. So if you dig into the literature a little bit, you find that as these seeds are getting planted, they're coated in a talc or graphite powder that creates a dust as they're getting planted in the ground, and the wind will blow this dust across the landscape, and it's laden with these neonicotinoid residues. And so we've seen other research that have they've set up little traps outside of field margins. This is a heat map showing where neonics were found after planting. And you can see that the dust gets blown out of the field, and we can find it, well outside the field onto non-target plants and potentially, flowering plants like dandelions, for example, and apple flowers that are all blooming during the same time period when these seeds are getting planted in the ground in May and June. And so concerns about dust had started a long time ago. In 1999, the dust was implicated in abnormal bee colony losses in a number of countries in Europe. And it was first implicated as a potential route to bee exposure like 20 years ago. We've been thinking about this dust as a problem for a long, long time. So we were thinking, well, let's look at apiaries that are near corn and soy crops. Apiaries are a bee yard. It's like a place where beekeepers keep all their honeybee hives. Let's look at sites that are near these corn and soybean crops, and then sites and apiaries that are not near corn and soybean crops. Because we know that the corn and soybean is the biggest use of neonics, so that would make sense that might be where they're getting exposed, right? And let's also look at when these seeds are getting planted in the ground, and then when they're not getting planted in the ground, to see if we see more exposure during those planting events as a result of that dust. And so in 2023, we tried to focus our sampling to kind of accommodate those hypotheses. And we focused sampling during and after planting, 29 apiaries with 17 with row crops present and 12 without row crops present. As I mentioned earlier, this is data I just got back a couple weeks ago, and we're doing more landscape level analyses coming up, but I don't have, I'm just doing with and without rather than looking at the percentage of row crop at this point. We had 61 bee collected pollen samples. So, up from 18, it's a huge bump in the number of samples. We finally got funding to do this. And then we also thought it'd be interesting to look at flowering plants out on the landscape to see not just the bees are bringing back, but can we find those neonics on plants that weren't necessarily treated that the bees or butterflies or other insects might be visiting. And I also looked a little bit at soil. And so similar to the data that I presented to you before, just an overview of all the different things we found. 61 bee collected plastic. We had 309 detections of 34 different pesticides. And if you look at all of the bee collected pollen samples, if we're looking just at neonic detections, almost 30%, so 29.5 of all of those samples came back positive for one of those neonicotinoids. Yeah.
So I'm a little confused as to which colonies are being tested here. Are these colonies that have failed or colonies that have not failed?
Colonies that have not failed, because we need the little bees workforce to be bringing back their
Right.
Yeah.
But I mean, I'm sure there's a period of time before they fail that they're performing that task.
Yes.
So have there been any tests on like the timing of these colonies that have failed, but we have evidence of what they were exposed to before they failed?
Elsewhere, there's a lot of work
Okay.
on that. So this was done in 2023. So I'm actually looking forward to talking to those beekeepers in the spring and saying, are your colonies still alive?
Right. Okay,
Yeah.
fair enough.
Yep.
Thank you.
Yeah, great question. So this is across all of the samples. Now, if you just isolate the colonies that were near row crops, how many of those apiaries are bringing back, or how many of those samples are bringing back neonicotinoids? And we find that 41%. So if you're a beekeeper and you're near row crops, 41% of those samples, we found that they were coming back with one of those, at least one, some more than that, one of those highly toxic neonicotinoids.
And row crops of corn and soy,
Pardon?
corn and soy,
Yes.
row crops. Yeah.
And this figure, I sorry I didn't explain it before, it might be a little confusing. So we did a little mapping exercise, just curious about how much corn and soy might these bees be coming in contact with. And so these circles represent an estimate of how far a honeybee might forage from their hive. So it's sort of like the flight zone. And so if you imagine if their little bee yard or their apiary, so where the hive lives, is in the centroid of these circles, like this sort of represents the amount of area that they could forage on across the landscape. And this is in Addison County. So these are 3 different apiaries of a commercial beekeeper in the state. And you can see all of the corn. This was some land cover data that was publicly available. All the corn was coded in red and the soy is purple. So you can see how much of that is within a flight zone of some of these apiaries. So we say, if they're near corn and soy, this might be what it looks like. But it could also, it can vary.
What's the diameter of sort of radius of a circle there?
1 mile is a radius, yep. So 2 mile, or no, wait, I'm sorry, 2 miles, 2 miles. Yep. Okay. Sure. So this is kind of another way of presenting the same data that I showed you earlier, or just that I wrote out, but it's much more visually, I think, helpful to see this. So these are color coded based on those 4 different neonics: thiamethoxam, clothianidin, imidacloprid, and acetamiprid, and that we found in our samples. This is the parts per billion. So this is not just whether or not it was there. This is the amount of that neonic that was detected in a given sample. And this is samples that were collected from sites with row crops present, and then sites without row crops present. And so you can see, again, like all of the hits for these neonics are on sites where the bees are foraging near row crops. And then if you're curious as to like, well, does this amount actually matter? Like 0.2 part per billion, like is that even significant? Is it biologically significant? So folks at Cornell did this big, awesome analysis looking at hundreds of different studies to get a better understanding of what the LOEC, the lowest observed effect concentration. So LOEC, it's abbreviation for that. And it means the smallest amount that's known to cause an impact on an organism. And they found so we're just going to, in this case, make it simple, just look at clothianidin here, which is the one that we found the most of. We got the most hits for clothianidin, right? At levels of 0.1, we see physiological impacts to honeybees. And I just, you see this threshold up a little bit. So at 0.9 part per billion, that's when studies are showing behavioral impacts to honeybees. So grooming behavior impacts, increased virus levels. So those viruses that are transmitted by the mite, this is an example of those synergistic impacts that the viruses are, the bee's immune system is suppressed, allowing for the viruses to replicate. So they have increased virus levels when they have, tiny amounts of exposure. And then jumping up here at 5.12 part per billion, that's where we expect to see some reproductive impacts. And studies found that queens were 34% less likely to survive. So many people know, like a honeybee colony consists of usually one queen and then lots of workers, and she's sort of the only one that's laying eggs, and all of her workforce are her daughters. And so if you have a problem with the queen, or a queen dies, that could result in the death of the colony. So queens are less likely to survive, 34%, and they have fewer eggs. All this to say is, yes, based on the data, these detections that we're finding here in Vermont honeybee hives are biologically significant. However, not however, but also, these things are really hard to measure. It's like really, you have to do a really significant study to go out and, find out, well, how likely is it for a queen to die? You'd have to go find the queen every day or count how many eggs she lays every day. And so without doing all these studies here in Vermont, we have to rely on some of the research that other people have found to understand what's the biological significance of this level. And so oftentimes when beekeepers are seeing problems with their hives, I think when people are thinking of pesticide exposure events, they think that a beekeeper is going to open the hive and they're going to see all the bees that are dead right away, like they've been exposed. And that can happen. That can surely happen. But with neonics, there's these sublethal impacts that cause these problems, physiologically, behavioral, reproductive, that kind of over time can create problems for a colony unless you're closely counting the number of eggs. A beekeeper might not notice it until it becomes more of a problem. You have a question here?
How do you find a queen?
Yep. So it can be more difficult when you have a bigger hive versus early in the season when the hive is a little bit smaller. She's easier to find. She looks different. Her abdomen's a little bit bigger. She walks different. It's kind of like a Where's Waldo situation, really. You pull up a frame and you try to find her. Lots of beekeepers will mark their queens with a paint marker on their thorax. And fun fact is that odd number every year is a different color. So you can tell how old your queen is based on the marking on your queen. And the interesting thing that I've talked to older beekeepers who've been at this for a long time is I think that there's a 5-year cycle where the colors change every 5 years because queens used to live for that long. But now you never see that color from 5 years ago because queens are only living 1 or 2 years and they're not superseding. So that color system is sort of in a lot of ways irrelevant. They should maybe have a 3-color system or a 2-color system because queens aren't living as long as, and talk to commercial beekeepers to hear that. And so as I mentioned before, we looked at neonicotinoid residues during when corn was being planted, and then in a period of time when corn and soybean was not being planted, so that May, June period, and then afterwards, and found that thiamethoxam and clothianidin in particular, all of those hits are happening during that planting window. So the data here suggests that this time of planting is a significant time when bees are getting exposed to these neonics. So a little bit about plants now, because remember I said we tested the plants. Okay, so corn is a plant that doesn't produce nectar. It's wind pollinated, so it doesn't need that sugary substance to attract pollinators or animals to move their pollen around. But they produce a lot of pollen. And I think it's often people often assume that bees will not visit corn because they don't produce nectar. This study was taken in Vermont this past summer. We found lots and lots of honeybees foraging on corn. And previous research has found that corn clothianidin concentrations in pollen can range from 1 to 6 parts per billion. We are tested the corn tassels, so that whole strand of lots of pollen, and found 1.79 to 7.53 parts per billion in those corn tassels. So it's possible that bees are out collecting corn pollen, and that's another exposure route outside of that dusting period. As I mentioned before, when these seeds are planted in the ground and 90% of the insecticide ends up in the soil and in waterways, neighboring plants can take up those residues from the soil, much like how neighboring plants take up nutrients from the soil. And those plants can then express it in their nectar and pollen. And so we tested flowering plants last summer and also some years ago too. But this past summer in 2023, we found 22, almost 23% of the flowering plant samples we found neonic residues on. And if you're curious about what plants we're finding neonic residues on, dandelions and apple tree blossoms, which I think, no, you're good. I think that those are that's probably a result of the dust because those are really early blooming flowers. And so as the dust gets blown, it lands on those dandelions and apple blossoms. And then we also found last year goldenrod, which is a really important source for honeybees that they collect for their winter stores. That's what goldenrod nectar is what they eat throughout the winter here in Vermont. And then also milkweed leaf tissue. We found thiamethoxam. That's the abbreviation for thiamethoxam. And we know that milkweed is the larval host plant for the monarch butterfly, right? And we think that I'm hypothesizing that when we're finding it in goldenrod and milkweed, that's the indication that it's probably getting taken up from the soil versus just the dust, because these are the early season blooming right when the dust is being generated, and this is, later season when at that point the dust is probably degraded by UV degradation, and it's probably in that case being taken up from the soil.
Sorry, is the assumption here that these are near row crops?
Yes, so the goldenrod was adjacent to row crops. These I collected from the roadside as I saw the tractor planting the seeds. So I saw the dust that day. And then milkweed leaves. This was actually taken from Borderview Farm, where many of you might know Heather Darby works, in a field that had been historically, I think, planted with corn years and years ago, and then had planted with milkweed. And so it was, 10.3 to 13.6 parts per billion in that milkweed tissue.
Residual from years prior.
Yes. Yeah. Should I continue? Or do you want to stop?
Yeah, no, we're just doing a time check. We've got about 10 minutes.
Sure.
So if you have more to share and
I have
wanna
a
keep
couple
going.
slides, just this part, and then this little section about. Okay,
Sure.
and then okay. So there's been a lot of research. So with all these issues with neonicotinoids and their impacts on target organisms like pollinators, there's a question of do farmers need this tool? Are they really effective for the crop? Because we don't want to take a tool away from the farmers. They're really important for crop yields. But the overwhelming evidence, and this is from a Cornell report from Scott McGarvey, he also presented this work to the Ag Innovation Board. So this whole talk you can view it online. They did an analysis looking at hundreds of different trials and found a very small benefit of using neonicotinoid treated seeds compared to untreated seeds or fungicide only. So 13% of the time it was beneficial. In this case, 11% of the time it was beneficial. But when you look at net income, so the economics of this, there was no difference in net income using these seeds or not using these seeds. So we have an instance where 99% of all these, this is corn specifically, 99% of corn is over 99% is being grown from these neonics, but it's actually unfortunately providing very little benefit. So if you look at the benefit cost of these insecticides, it doesn't really make sense. So this was some unpublished work from Heather Darby. I'm sure you're going to be hearing from her. She presented to the Ag Innovation Board. She did these trials up in Aalborg. It's part of the first year of a 2-year study. And she again found no significant differences across treated and untreated seeds in her trial plots. That's for corn. For soybean, the EPA, you can use this little QR code and just look up. The EPA concluded that the seed treatments provide little to no overall benefits for soybean production in most situations. Just right on their website. And so with this bill getting passed into law in New York, and then you may be aware of in Quebec, our neighbors to the north, they started limiting neonicotinoid treated seeds in 2019. And so those farmers have had a lot of time to transition and to adjust. And so we thought it would be really interesting to have the Quebec farmers who have faced the challenges of trying to adjust from these restrictions on neonicotinoid seeds. How are they growing? What are their row crops looking like now? Were they able to source non-treated seed? Because that's an argument we keep hearing. Farmers just can't get non-treated seed. It's an argument we keep hearing. So we held a farmer panel discussion just this past week, a couple days ago, where we had a panel of 4 Quebec farmers. We interviewed them. We had a series of 4 questions and asked them about their experiences. Can they find non-treated seed? What are they seeing in differences of row crops and all of these things? And so it was reported, and I recommend if you guys have the opportunity to watch that. Some of the highlights is that they went from 99% corn and 50% soybean being treated to 0.5 of corn and none of soybean, so this was really effective. They are saying that they're seeing no impacts to crop yields as a result of making this switch. We would think that we would be hearing about it if it was devastating their crops. So they said that that there was a lot of fear. Farmers were very fearful of not binding the seeds, but they said that the seed companies reacted, and now they not only supply non-neonic treated seeds, but they're actually supplying non-insecticide treated seeds altogether, because neonics aren't the only thing that can be used on these seed treatments. While some farmers moved to some of these alternatives, diamides, which you'll probably hear about, others have abandoned insecticide treatments altogether. So those are some of the takeaways of that Quebec's farmer panel discussion that I held in conjunction with Heather Darby from UVM Extension just past week. And that's it.
Just diamide, go back to that for a moment.
Sure.
That's an existing, it's not a novel or new product that may have unintended consequences that we'll discover 20 years from now, or?
So the data show that they are really bad for Lepidoptera, so moths and butterflies, but they're not as toxic to bees. So it's not great. Heather was asked by some folks if Vermont farmers are using diamides right now, and she said no. And I reached out to the Ministry of, I guess, our equivalent of our ag, food and markets in Quebec. Sorry, and asked about this, and they said by 2025 they're planning to phase out all insecticides on treated seeds, because they did see a switch to these diamides, and now they're saying, well, let's just get rid of them altogether. So it was interesting.
Representative Lipsky.
When you cross the border from the USA up into Quebec at, say, Highgate, you get the opportunity to see vast sort of floodplain ag. Where were the locations of these 4 farmers? Were they adjacent to Vermont, or were they all over the province?
These are, you guys can't read them. I'm unfortunately not super familiar with Canadian geography, but their locations are listed here, and I can try my best in French to pronounce that, or you can.
Okay.
Sure.
Representative Templeman.
Okay, so in your studies of productivity of the farm, with and without the seed treatment, I'm wondering, in the cases where seeds were not treated, what were the methodologies employed to prevent pest problems? So, I mean, was it 0? Are we comparing to a no pesticide regime? Or are we comparing to multiple regimes that are to make up for there not being the neonicotinoids in the seed?
Okay, so just to be clear, I didn't do that work.
Okay, fair enough.
But in this case, they looked at neonics compared to untreated, so nothing.
Okay.
Then these are non-insecticide treatments, which really just means fungicide only.
Okay.
This is seeds treated with a different kind of insecticide.
Right.
And these are insecticides applied to the soil, not planting. So they looked at various different methods of either not using it at all and then using it in different ways. And then you would have to ask Heather directly about the I'm not sure. I think that she tried to keep all the variables the same, and the only difference my assumption would be that she just did the seed treatments would be the difference between those two.
And then regarding the farm that Heather Darby, so this one, with the high levels in older fields, I mean, clearly there's a residual and a lasting effect. Have we seen where neonicotinoids have been outlawed or banned? Do we see an increase in viability for bee colonies, or is it too early to say because the persistence of these chemicals?
Yes, my understanding is too early to say because of the persistence of the chemicals. Yeah. And you touched on a point about persistence in the soil, and the best example of this I've seen in all of the literature is actually this data from Heather's work here in Alberg, looking at persistence in the soil. But these are actually, okay, so these are fields that have never historically been planted with neonics. And at a depth of 0 to 2.5 inches, she didn't find any clothianidin. But at a depth of just a little bit below this, that's what you'd expect because of UV radiation, right? UV degradation. At a little bit below the surface, 2.5 to 6 inches, 75% of those samples were positive for neonics at a level of 6 parts per billion. So you think about where 6 falls on that.
And these were never treated.
These are fields that have no historic, no historic.
So where is it coming from?
Great question.
Adjacent fields?
So these data really surprised Heather, and she can speak more to that, but we discussed perhaps, so because they're super water soluble and can travel really long distances, they can move subsurface in the water table underneath the ground. And then also maybe the dust that can land outside and then maybe go, get washed into the soil, but I don't know at this point.
Yeah, let's take one more question, Representative.
So I was just wondering if the trials have been over multiple years or they're just 1 year trials.
Heather's trials are just 1 year. And it is a part of a 2-year study, but the hundreds of trials that were done by, that were analyzed by Scott Nicart, those were multi-year studies that looked at differences in benefits to crop yields.
Okay, well, that was an awful lot.
I know.
And I think the way you presented it though was very helpful. And I know there was more that I saw that you had possibly some other slides.
Those were like, if anyone had a question, I could.
Okay, sorry. All right, well, as we dig into this topic, we may reach out
Sure.
to have you brief us again. So thank you very much.
Sure, thank you for having me.